Under techniques, today we are going to talk about agarose gel electrophoresis. It is a molecular biology technique widely used today, and it is used mainly in research work as well as in laboratories. If we define electrophoresis, it is a technique that separates large charged molecules such as DNA, RNA and protein according to their molecular weight. So it is happening in a gel actually. You have to form a gel first. In these molecules there is a charge. So the migration of a molecule is depending on the net charge and its size. In gel electrophoresis, molecules are moving in small pores of the gel matrix. When the gel is formed, the small pores are made and the molecules are moving in them. The pores are restricting the movement of the gel. So when it is moved, because of this restriction, it helps in the separation of gel. So according to the molecular weight, because of the pores, the movement is restricted and the molecules are moving with a restriction and they separate according to their molecular weight. Usually larger molecules move slowly and smaller molecules are moving in a fastly manner. And since there is a charge, the molecular charge is very important. The charge is depending on the length. Because of this, the separation is based on the size also. So, for the separation of DNA, agarose gel electrophoresis is the most frequently used method. So, this is a agarose gel which has run. So we are going to see how the gel is going to run. The gel is made with the powder. It is known as agarose powder and it is purified agar. It is not having any impurities. And when it is formed, it forms a polysaccharide matrix. And there are other gels also. One is polyacrylamide. The agarose is the most common. Agarose is obtained by the red seaweed and actually agarose is used as a key ingredient in commercial ice creams and jellies also, but those are not purified products. The purified agarose is getting to research work. And polyacrylamide is also used as a gel base, but it is used for, it is forming small pores and it is used for small molecules. And agarose is used for larger molecules such as DNA. When the agarose gel is formed, the matrix looks like this. This is 1% agarose. You can see the pores are very small and those small pore size is having low concentration and it is good for resolution of DNA molecules, and this is 2% agarose and it is having much more smaller pore size. It is having high concentration. When it is become very small pores, it is very good for very small molecules such as DNA fragments. DNA is a larger molecule, but in research work we need to make into pieces. So those DNA fragments are separated by high percentage agarose gels. So our overview of this lesson is gel preparation, loading the gel and running the gel. So we learnt about what is agarose. Now we are going to see how the gel is prepared. Gel preparation is done in the laboratory using agarose powder. It should be dissolved. To dissolve it, it should be heated with the buffer in a microwave oven. So like a normal gel, it is formed, and when it is dissolved, it should be poured into a container. We can't pour into a normal container. There is a special equipment known as gel casting tray, and this is a gel casting tray. And to load the gel, to load the samples, there should be some pores. To form the pores, gel comb is inserted. Here you can see small wells are there, but the gel comb is not there. It is having just like a comb, it is inserted before the gel is solidified. So this is the overview of the first step. So this is gel is dissolved in the microwave oven, it is poured into the gel casting tray, and the comb is inserted here, and the wells are formed here, and we have to allow it to solidify for about 20 to half an hour. And when we load the DNA sample, it should be visualized. So we should stain the gel. To stain the gel, we have to add some stains. Most commonly used stain is ethylene bromide, but it is carcinogenic. We have to take precautions to minimize the damages caused by it. But it should be a small drop of ethylene bromide should be mixed with the gel before solidifying and before pouring to the before solidifying the gel, and it is poured, and then the stain is there in the gel. So this stain cannot use to differentiate the DNA. For differentiate the DNA, we have to run with a marker ladder or a DNA probe. This is just to see whether DNA is present or not. So this is just a stain to visualize it. When we are loading the gel, we have to use a micropipette, and we have to load right into the well. And this is gel is almost transparent, and if our sample is also transparent, we can't see it. So we are using gel loading dye into it. It is not ethylene bromide, it is something else. So it is having in blue color as well as green in color. So you can see the color to load the sample easily to the well. So now our gel is ready. We are going to run the gel. To run the gel, we are using agarosteric electrophoresis equipment, and it is an equipment having 2 electrodes. It is like a tank, and it has negative and positive electrodes connected to the power supply. So you have to pour the buffer and put the gel in the middle, and then you have to connect the equipment into the power supply and allow to run at least half an hour to 1 hour, depending on the type of the DNA, whether it is whole DNA or DNA fragments. So we allow it to run, and when it is running, the negatively charged DNA molecules migrates towards the well. So here is the well, and the migration occurs from negative electrode to positive electrode. So it moves towards the anode. After running the gel, we can see the loading dye has moved. We can't see any DNA there. To visualize it, we are using UV trans illuminator. When we are using the UV trans illuminator, we are exposing the gel into UV light, and you can see the gel like this. These are the wells, and here the DNA samples have run to the bottom. So when the gel is run, we are going to analyze it. we have to analyze it. I mentioned we have to use a Mahatlanda for this, and this is the Mahatlanda, and it is having the molecular weight of the DNA fragments. Usually Mahatlandas are coming commercially with the known molecular weights. These are our samples, 1, 2, 3, 4, those are our samples. And you can see in lane number 1 there is no sample, and it is using acid control usually without DNA to see whether there are impurities. And in lane number 2 there is a DNA fragment, the molecular weight is 1500 base pairs. And in the third lane there is another one with 650 base pairs molecular weight, and fourth lane has the smallest one, 4000 base pairs molecular weight. So you can see, depending on the molecular weight, the length of the movement is different. So we are going to our next molecular technique, polymerase chain reaction, which is talking these days a lot. This is also a technique in my molecular biology. When we test the samples, actually here in all these techniques we are dealing with nucleic acids, DNA or RNA. So in polymerase chain reaction or PCR also we are dealing with DNA samples. If you see the history of this technique, it was invented by an American biochemist, Kary Mullis, and he was the Nobel Prize winner in 1993, and he awarded with many awards such as Japan Award, and he developed this technique. It is a rapid method of obtaining large number of copies of DNA at high accuracy. So in this method actually we used to copy the sequences of DNA in vitro. It means in experimental conditions, because in vivo, it means in live conditions, DNA copying is happening in our bodies even now in each cell of us. So it is a natural process. So mimicking that, we have developed a laboratory technique to copy the sequences of DNA in the laboratory or in vitro using the mechanism of DNA replication. This method was developed by the scientist, and it is used in laboratories and research work and in most of the scientific work today. PCR is occurred inside a PCR machine. Technically it is called thermal cycler. So if you see the details of the thermal cycler, it is having a heating block, upper and lower. Here in the lower block we are inserting the samples, and we have to close the lid. With the upper heating block there is a lid here, and the display is there. Because this is an automated machine, it means actually this is a chemical reaction, polymerase chain reaction. It is a cyclic reaction. There is a need for temperature changes in this reaction. So this machine allows to change the temperature according to the reaction. PCR is done in PCR tubes. There is a PCR mix we have to insert into the machine. In the PCR mix, the key ingredient is DNA polymerase enzymes, which is occurring naturally in our body. But natural DNA polymerase enzyme is usually it is inside the living cell and it is depending on our body temperature. Here the temperature changes are very high. So a type of DNA polymerase enzyme is used here. And other than that, here we are going to copy DNA. For copying there should be materials. So there should be DNTPs. It means deoxyribonucleotide triphosphates. Because there are 4 types of nitrogenous bases in DNA, there are 4 types of DNTPs. DATP, DGTP, DTTP and DCTP. And we are going to copy something, so there should be a DNA template to be copied. So single-stranded DNA template is necessary for the PCR mix, and also to start copying there should be a primer. In replication mechanism you have learnt about primers. Usually primers are RNA sequences, but here primer is a DNA sequence. It is a short sequence and it is necessary to initiate the mechanism. And also some magnesium ions are also needed. So this is a close view of the PCR machine. So the tubes are inserted into the heater block here, and this is how a tube is inserted into the heater block. And this is what's happening inside the PCR machine when you mix these components of the PCR mix and put into the thermal cycler. The reaction undergoes is known as the PCR process. This is one cycle. These are the temperature changes. There are actually 3 temperature ranges depending on the phases, and there are 3 phases in the reaction. I told you this is a cyclic reaction, and temperature changes from 95 degrees Celsius to 55 Celsius degrees and then into 72 Celsius temperature. And these are the stages in the detailed manner. First stage is denaturing stage. It means double-stranded DNA is denatured into single-stranded. The next one is the annealing stage. The temperature is 50 to 56. This temperature is dropped there, and the primer is annealed there. That's why it is annealing stage. And this is extending stage, and the temperature again rises to 70 to 72 degrees of Celsius, and new strand is copying at this stage. So this is how the reaction occurs in the cyclic manner. There are many cycles occurring in one PCR process. If you go to details of this, in the denaturation stage, you can see the temperature difference. It is heated to 95 degrees of Celsius. Usually DNA is denatured at this temperature. It is almost close to the boiling point of water. So you may wonder how enzymes are existing in this temperature. I told you it is not normal DNA polymerase, it is a type of DNA polymerase. So that DNA polymerase can work out here, and that DNA polymerase is known as Taq DNA polymerase. Taq DNA polymerase is obtained by a special microorganism. Actually it is a bacteria, Thermus aquaticus, and it is thermophilic bacteria, and it is living in, it is something elongated rod-shaped bacteria, and they live in volcanic peaks. and sometimes in the seabed also there are some hydrothermal vents and hot water springs where the temperature is very high. When it comes to the surface of the land, the temperature is dropped little bit, but all these volcanic peaks and hot water springs usually have a higher temperature at the bottom because it is connected to the core of the earth. So the temperature is 50 degrees of Celsius to 80 degrees of Celsius. These bacteria can survive in that. So when we go to the annealing stage, the second stage, the temperature is dropped here from 95 to 55 degrees of Celsius. So what happens there? The primer is annealing. You can see the double-stranded DNA is separated now. The primers are annealing here, and it has this annealing temperature, and primers are binding at this temperature. And the extending stage is the third stage, and in that temperature is rising to 74 degrees of Celsius, and now the primer has started copying the new strand, and the new strand is synthesized there. It means the DNA is copied there. So this is the process, and it is a longer process, and usually when it happens in the first cycle, after the first cycle, another set of copy is formed, but this copy is having some problems. It is a bit longer. It has, because of the primer, it has some extra nucleotides, so it is longer. So after another couple of PCR cycle, it becomes normal. After, it means after the third cycle, the PCR process is increasing. The copying is increasing in an exponential manner. It means 2, 4, 8, 16, etc. And there are about 35 to 40 cycles in the PCR process, and at the end, millions of copies are formed. So this is showing how it is grown exponentially, and you can see this is the first cycle. The DNA copy is a bit longer. In the second cycle also it is similar, and in the third cycle only the exact copy is obtained. And here it shows how it is increasing, cycle 1, 2 and 3 exponentially. So when we go to the applications of PCR, PCR is used in many processes which we are using. Actually it was used widely for research work, in genetic research. Even today it is used in genetic research a lot. Other than that, it is used in environmental biology, medicine, phylogenetics and forensics, food and agriculture and consumer genomics. So if we discussed in detail, today we are using most clinical applications using PCR. For example, hepatitis, HIV and also coronavirus. And other than that, genetic diseases. Sometimes cancer is also a genetic disease, but there are some more genetic diseases such as cystic fibrosis and sickle cell anemia, which is simulated thalassemia in Sri Lanka, and also phenylketonuria. And it is used in forensics also. In forensics, there is something called DNA fingerprinting. sample of a victim in the crime scene, we can identify the criminal using PCR if we can get a small sample such as a drop of blood or a single hair even. We can identify the criminal using PCR, and it is an essential technique in cloning procedure. DNA cloning is done to obtain some genetically modified organisms such as some agricultural products which are resisting for a long time or giving high productivity. So cloning procedure is using the technique of PCR, and there's another molecular technique, DNA sequencing, and it depends on PCR. DNA sequencing is genotyping. It means typing the nucleotides. For that, lot of PCR copies are necessary. If we have a small sample, we can increase it using PCR, and it is also used to identify and explore relationships among species in evolutionary biology. To study especially some samples, as in Jurassic Park, some preserved samples, very small amount is there. We can copy it using PCR and study in the evolutionary biology. And there is another subject area called anthropology. It is the ancient human migration. In that case also the samples are limited, such as some skeletons are there. So those samples can be increased by using PCR and using studies. And it is also another related subject area to anthropology, paleontology. It is used DNA amplifications to study about the species and how the species are related using preserved fossils of millions and billions years old. So those are the common applications of PCR. And this is my question. So I want you to write a short note on agarose gel electrophoresis, polymerase chain reaction, and also DNA analysis. And thank you for listening.